For millions of people around the world, pain isn't just an occasional problem, it's a constant companion. Chronic pain is one of the biggest medical challenges of our time, and for years, doctors leaned heavily on opioid drugs, powerful but dangerously addictive. In the middle of this crisis, researchers began looking for alternatives. One unlikely place they turned was scorpion venom. In preclinical trials, venom-derived peptides are first tested in cell cultures and animal models. Can they block pain signals? Do they affect breathing, blood pressure, or other vital systems? Some peptides show a remarkable ability to selectively block just one type of sodium channel, NaV1.7, a key player in pain transmission. People born with mutations in this channel can't feel pain at all. Venom gives us a way to dial that effect up or down temporarily. If a compound looks promising and safe enough, it moves into human trials. Phase 1 trials focus mainly on safety. Small doses, carefully monitored. Phase 2 and 3 trials ask a different question. Does this actually help real patients? Imagine a patient with nerve pain so severe, even a light touch feels like fire. For some of these patients, early venom-based treatments have reduced pain without the foggy, addictive side effects of opioids. These aren't magic bullets. Not every patient responds, but the results are strong enough that pharmaceutical companies are investing heavily in venom-inspired pain therapies. Behind every data point on a graph is a human being, hoping that a molecule from a scorpion will finally quiet the signals of pain. Turning a raw venom peptide into an approved drug is a marathon, not a sprint. Sometimes 10 to 15 years and hundreds of millions of dollars. Step 1 is discovery. Scientists identify a peptide with an interesting effect, like blocking a specific ion channel. Step 2 is optimization. The natural peptide is often unstable. It might break down too fast in the body or trigger immune reactions. Chemists tweak the molecule, changing a few amino acids, making it more stable, less toxic, better targeted. The goal is to keep the useful effect and minimize the risks. Then come extensive safety tests in animals. How does the compound behave in the liver, kidneys, heart, brain? At what dose does it become dangerous? Only when that window looks wide enough does the drug move into human trials. Most candidates fail somewhere along this path. But even failures teach scientists something. Every dead end refines our understanding of how venom molecules interact with the human body. If a candidate survives all 3 phases of clinical testing, regulators review thousands of pages of data. Only then, if the benefits clearly outweigh the risks, can a venom-derived drug enter the market. By the time a scorpion-inspired medicine reaches this shelf, it has been transformed again and again, from wild toxin to carefully controlled tool. As demand for venom-based research grows, technology is changing how we harvest and study these toxins. Some modern facilities are experimenting with automated extraction systems. Robotic arms can restrain scorpions with consistent gentleness, apply electric pulses with exact timing and voltage, and collect droplets with microliter precision. For science, consistency is everything. The more standardized the milking process, the easier it is to compare venom batches and design reliable experiments. Sensors can measure the exact volume of each droplet, its electrical properties, even its protein concentration in real time. All of this data feeds into large databases, tracking how age, diet, temperature, and species affect venom quality. Machine learning models can then predict which conditions produce the best venom for a given medical target, which individual scorpions are the most reliable long-term producers. In the future, farms may rely less on mass numbers of animals and more on smaller, carefully selected populations supported by precise automation. The same AI tools we use to design new computer chips are now helping us understand the chemistry of a creature that evolved 400 million years ago. Pain and heart disease aren't the only frontiers for scorpion venom. Some peptides have a surprising talent. They can home in on cancer cells. Certain venom components recognize molecules that are overexpressed on the surface of tumor cells. That means we can use them like tiny guided missiles. Scientists have attached fluorescent dyes to chlorotoxin-like peptides. When injected, they highlight malignant cells under special cameras, helping surgeons see the true borders of a tumor during surgery. In brain surgery, where every millimeter matters, that extra visibility can mean removing more of the cancer while sparing healthy tissue and function. Other projects are exploring direct anti-cancer effects. Can venom-derived molecules slow tumor growth or make cancer cells more vulnerable to existing chemotherapy? Most of this work is still early stage, but it shows how a single venom can have many different applications, not just blocking pain, but also revealing and attacking disease. As venom-based drugs advance, new questions appear. Who will actually have access to these high-tech treatments? Will they remain expensive niche therapies or become widely available? Some of the deadliest scorpions live in poorer regions where antivenom is already hard to get. Ironically, the same countries that suffer from stings might not be the first to receive the most advanced venom-derived medicines. Collaborations between universities, governments, and companies are trying to change that by sharing data, technology, and training so that venom doesn't just become another way to widen the global health gap. There's also the question of animal welfare. Scorpions are not cuddly mammals, but they are living creatures. Non-lethal extraction methods, reduced handling, and better habitat design are all ways to respect that fact. In the end, the story of scorpion venom is a story about how we choose to use knowledge. We can see these animals as monsters to fear, or as partners in a quiet revolution in medicine. From their ancient armor and stingers to modern robots and hospital labs, scorpions show us that nature's most dangerous designs can, with enough care, become tools for healing. If you enjoyed this deep, long-form exploration of venom in medicine, subscribe to BioInspire Tech for more human and nature-inspired technologies. Tell me in the comments, should the next full documentary be on snake venom drugs, cone snail neurotoxins, or jellyfish toxins? Your suggestion might shape the next episode. Thank you for watching and for seeing the hidden technology inside nature.